All right, welcome back. Today we're going to be talking about DNA replication. So DNA replication is making copies of DNA. To replicate means to make a copy. So we're talking about how we copy our DNA. Now remember, reasoning behind copying our DNA or repair damaged things. So I cut myself, making new cells, copy DNA in order to make a new cell. So in our last video, we talked about how if I have one side of the DNA molecule, I can figure out the other side because I know A and T always go together and G and C always go together. Therefore, that base pairing rule of A goes with T and G goes with C will allow me to use each side of the DNA as a template to make a new strand. So if I know one side of the DNA, I can figure out the other, and then that can be used to make a completely new strand of DNA. In this process, we're going to have several enzymes that are going to be involved. The first enzyme is DNA helicase. DNA helicase is going to unwind or unzip the helix so that the DNA can be copied. The way I remember this is DNA helicase unzips your genes because it takes the DNA and splits it right in half. The second enzyme that's going to help out is called DNA polymerase. DNA polymerase is going to bring in the new nucleotides and proofread in the end. So the way that I remember that is P for proofread, P for polymerase, build and proofread. And then the last enzyme that's going to help out is called DNA ligase. And DNA ligase is going to glue the new pieces of DNA together to form one continuous strand. So we're going to go through this in a bit more detail. So the first step when I'm making a copy of my DNA, the first step is going to be to unwind the helix. So helicase unzips the helix or unwinds the DNA. And this is going to create, if you look over here, if I unwind a helix right in the middle, it's going to create a little bubble that's called a replication fork. And the replication fork is where the DNA is split so that it can be copied. So after I unzip the helix, the next step is going to be to add bases and make the new side of DNA. And DNA polymerase is going to bring in those new bases. And DNA is anti-parallel, meaning one side is right side up and the other is upside down. So if I look at this diagram over here of DNA, this side is right side up, the other side is upside down. So you're going to want to remember that. DNA anti-parallel, one side right side up, the other side upside down. And that's going to make it possible for the bases to join together in the middle. Each side of the DNA has a name. The side with the phosphate is the 5' end. The side with the sugar is the 3' end. So phosphate 5. Phosphate 5, sugar 3. And that's just the direction of the DNA. And when copying DNA, it's always copied in the 5-3 direction, meaning P for 5, the sugar is 3, it always reads this direction. So one side of the DNA is red one direction, the other side is red in the other direction. Just catching up. So the end result of DNA replication, after this happens all the way up and down both sides, is going to be 2 DNA molecules that are identical to one another. Each new DNA molecule has one strand that came from the original DNA and one strand that is new, and that process is called being semi-conservative, meaning one side is old and one side is new. So semi-conservative, one side old, one side new. Now, sometimes in this process, errors occur. If you do something thousands, millions of times in a row, sometimes an error is going to occur. And when an error occurs, sometimes it can be caught before it becomes permanent. And that's the job of DNA polymerase. DNA polymerase is going to check behind itself and proofread and then try to correct any mistakes that are made. So any mistakes that are made, DNA polymerase is going to try to find and fix right there when it's making copies of the DNA so that there are no mutations in the end. All right, so that is DNA replication. I want to go through one little drawing quickly before we wrap up. So here is, I'm going to represent red is going to be my original DNA. And I'm going to use green as my new DNA. So when I want to make a copy of my DNA, it's all coiled up. First step is to uncoil the DNA. Once the DNA is uncoiled, these segments, right now I'm drawing them as just lines, but all along here, remember that these are carrying letters. So each side is carrying letters. So DNA helicase unzips. My sides have letters. What's going to happen now is DNA polymerase is going to come in and build the new DNA. So what it's going to say, it's going to run down in this direction. It's going to say, I see an A, and it's going to put a T. It sees a T, it's going to put an A. It sees a G, it's going to put a C. If it sees a C, it's going to put a G. And if it sees an A, it's going to put a T. And it does the same thing on the other side, except it starts at the bottom. If it sees a T, it'll put an A. If it sees a G, it'll put a C. If it sees a C, it's going to put a G. That's an A, so I'm going to put a T. And I see a T, so now I'm going to put an A. And again, I represented this with just a straight line. So now I'm going to be left with 2 DNAs. One side has the old DNA and one side has the new DNA. And before we wrap up, it's going to coil back together. And each side, one side old, one side new, therefore this process is semi-conservative. So this is what this process would look like. All right, so there's going to be some practice. Go ahead, check Canvas.